Okay, so good afternoon from me as well to everyone. I would also like to thank the organizers for inviting me this year again. It's always a great pleasure to be here, to feel this nice atmosphere and to learn about all the exciting research that you guys are doing. So hopefully today you're learning useful statistical and data normalization and analysis tricks that will help your own research. So far, essentially what you saw is the initial data preparation part. And I would say this is really the essential part. You always need to first look at your data, look at those distributions, really check if there's anything strange in there, find outliers, and also apply any normalization if it is really necessary, because if you don't set the stage in the right way, then all the analyses that you do afterwards may actually lead you to the wrong conclusion. So this is really the heavy lifting that you did till this moment. So now there will be a lot of fun things to come. There was one question that was about the multi-scatter plot, and if one can look at the correlations simultaneously, so it's actually possible. You see here, this is the multi-scatter plot that is somewhere at matrix 14 in this case. I don't know where it is for you, but it's this icon here. And then the way to display all the correlations is to go to this panel here, where you see you have many different options. So the default one is none, which is why you don't see anything by default, but then you can select any of this. And in this case, it's just Pearson correlation, and then you see, then it's shown for all the replicates. And then, as you would expect quite nicely around the diagonal, you see the biological replicates here with really narrow distributions, and then you see these would have then the highest correlations of the between replicate correlations. Okay, just a side remark. And also there was another question before that regarding the annotation files and how often are those updated. Probably not as often as you would want to, so it's not every week or every month. But what you can actually do is all these annotations, they come from UniProt, which is really an amazing resource for proteins. And any of you can just go to the UniProt website and then download these annotations, and even more for, not just for human, but for any organism that UniProt supports. And all you need to do is just download it from there and copy it in this conf annotation folder that Tommy showed you. So, and Perseus would find it and it would work. So you actually don't even need to wait for official releases. You can just download the files yourselves. Right. So we'll continue from where Tammy left, which will be the last matrix, the one where she saw that all the histograms are quite nice. So if it looks like this for you and you don't see all the histograms, you can easily fit them to the screen just by clicking fit width and fit height. So you see that they look okay. So there is no distribution that is really drastically different from the rest, which means that we can just go ahead and then continue with this data. So what we want to do next is actually to do some statistical analysis, and in particular, we're maybe interested in finding what are proteins that are differentially expressed between our 3 cell lines. So in your case, this can be disease and healthy groups or any other groups. But in order to start applying some statistics, first we need to tell the software what is our experimental design, or what are the biological conditions that we would want to compare. So to do this, we need to go to annotation rows and click categorical annotation rows. So you will see then this pop-up window where we can define the experimental design. So we can give it a specific name that is something meaningful to us. In this case, we have different cell lines, so just call it cell lines. And then what is important is that in order for the software to know that several samples belong to the same group, they should just have the same name here, so the same group name. And we can do this manually by just removing the _1_2_3. So you see now the first three samples from this group, they'll just have the same group name. Or if you named your samples in a convenient way and in following a particular pattern, so you see always that there's the name of the cell line and then underscore the number of the replicate, we can again take advantage of this of this regular expression feature. So instead we can choose here create group from experiment name, the second option. And here you see that there are already some patterns that are pre-coded in the software. So exactly this pattern where we have underscore and then some number is automatically recognized by the software. There are more options here. So there are more pre-configured patterns, for example, for LFQ intensity underscore, or if the name of the column would be normalized ratio HL, again with the underscores. Alternatively, we can define our own regular expression if the pattern that we want to match is very different, but we'll just stick to the first one because it already applies to our naming. So we'll just click OK. And what you see here is that there is a new row that has appeared here. So this is why it's actually called a notation row, because it's not part of our main matrix, but it appears here. And then you see that the first three replicates have the same name. So in this case, the software knows that we have three groups, and then it knows exactly which samples belong to each group. We can define many groupings, so we can just add another one if we wanted to. For example, if we would have biological and technical replicates, we would have to define those as separate groups. But in this case, we don't have this, so we don't need to add anything else. Okay, so now we're ready to do some statistical analysis. There is a menu here called tests. And you see quite a number of different tests. So we could do one sample test when there is just one sample. So I just ask, what if we had just one cell lines but with multiple replicates? Then this would be the right option to use. Again, only in case where you have multiple replicates. It won't work if you have just one sample. Then two sample tests, as the name implies, should be used when we're comparing two groups or two conditions. In this case, we want to compare three cell lines, so we're going to go for multiple sample tests. So this applies to everything that is more than two, 3, and so on. Okay, and then there are some more options for more complex designs. For example, if we have two factors, so we're not interested, we're not looking only at cell lines, but we would have more conditions, let's say cell lines in, I don't know, in different time points, then we can add a second factor and then Tyanova would be applicable. And maybe we go into post hoc tests, we'll see if we have time for this. So in this case, we go for multiple sample tests because of the three groups. And now you see the parameters of this test. So the first one is the grouping, so the experimental design. And here the software will offer you automatically the annotation rows that you have defined previously. In this case, we have just one called grouping, so we can't even select anything else. In terms of tests, there's just one also in this case, it's a novo. And here you see the S0 parameter that Jürgen described in the theoretical part before lunch. So another really important parameter is the how will our significant hits be defined. You remember the problem with the multiple hypothesis testing that we run into. So we need to indicate how we're going to control for this problem. And you also remember that what is recommended by the software is this permutation-based FDR. So in this case, you could choose not to apply any false discovery rate corrections, just use the p-values. You can use the Benjamini-Holberg FDR, but you remember that actually we avoid violating a lot of the requirements for the classical statistic test if we just use this permutation-based FDR. So this is the default one, this is the recommended one, but if you still want, you can change this. This would be the threshold that will be used, so everything with Q-value smaller than 5% will be marked as significant in the output table. You can, of course, change this, but you could also control this later in the table, so you can just keep 5%. This is the parameter that controls the number of randomizations. So you remember that for this permutation-based FDR, we create this background distribution. So here, the minimum that is recommended is 215. If needed, you can increase this, but you can also stick with this number. There is a parameter here, preserve grouping in randomizations. And this becomes important if you would have technical replicates that for some reason you do not want to average. So probably the easiest way is if you average technical replicates, but if you do not and if you keep them in this analysis, you use the permutation-based FDR, you would have to indicate them here. And as I mentioned, if you have technical replicates, you have to define another annotation row, as we did for the original groupings, and you would have to select it here. So it would be available here, and you have to indicate it here. And this is really essential with this way of doing the FDR, because otherwise you would create the wrong background distributions and you're going to end up with wrong statistics. So also really do not cheat. If you do not have biological replicates, but only technical replicates, do not use this test, because you're going to end up with a lot of significant hits that are just not true. All right. Here, there's the last thing you could change is suffix, in which case there would just be the suffix added to the output column name. I don't know. We'll just leave it in this case. But because if this is useful if you're running many different tests and then you want to keep track of which one this was. So it just adds a name to the output column. But we don't change anything here. We just continue. Click OK. So there is the output table already. What we can find in the output table are 3 new columns. So the first one is ANOVA significant, which is of type category. And what you would see is that there are pluses opposite to some of the proteins, of the protein groups. And if you actually click on the column gets sorted. So if you click twice, all the significant hits will then come on top. If you scroll, you can actually see that there are quite a few proteins that are differentially expressed between the 3 groups. Okay, a bit more than 2,000. So the other 2 columns that are produced as a result of the test are the minus log p-value here and the corrected p-value or the q-value that is here. So everything, essentially, you can also use this to trim down your significance level. So we chose 5%, but you could also sort these ones, and then if you want a smaller FDR, let's say 1%, you could also just filter it at a lower number, say everything that is smaller than 1%. That's why I said that defining the alpha level at the first step is not really essential, because here you could still see what are the most significant hits in your analysis. Okay, so the next thing one can do is you can, of course, just go and look at the differentially expressed proteins and then already start to make some biological rationale behind what is really going on or why are these differences there. Of course, it's interesting to see what is happening on the systems level. So are there any pathways or biological processes that are globally different between the different cell lines? And so for this step, it's really important that you all got your annotations, the Go terms and the KEGG terms, because we'll check if anything is enriched in here. So there are different ways to do this enrichment. One straightforward way is just to look at what is enriched among all of our significant hits. So to do this, we can just go to annotation columns here and select the Fisher exact test. This is also something that Jürgen described. So there, the first thing is we'll have to define where do we want to check for enrichment. And we said that we're actually interested in looking at what is enriched among our significant hits. So we'll select here the ANOVA significant column. So this is the column where we had all the differentially expressed proteins marked with plus. Here again, because essentially we'll be checking if any of our pathways or any of our biological process is enriched, it means that we'll be doing really many statistical tests. And we have the same problem that we'll again try to compute p-values, and some of those will be significant by chance. So we again need to control for this false discovery rate. In this case, it would be super difficult to do permutation-based FDR, so we stick to the Benjamini-Holberg FDR. There is a threshold here, which is kind of empirically defined, but really nothing stops you from setting this to 20% or to 5% or to any value, as long as you know what this value is and you report it later, right? And you know that this means that you expect that with this threshold value, at least 2% of your hits in the end will be falsely identified or will be falsely called significant. And relative enrichment we'll leave out for today, and I'll talk about it tomorrow. Okay, so I can just click OK. And this is a step that generally takes a bit of time because, as I said, really everything is tested in the background, so all the categories that we added. Now, maybe here I can mention that this is not the best possible way to do the enrichment. Now I'll explain why in a second. So this is already the output table of the Fisher's exact test. And you see it's exactly what, again, Jürgen showed on the slide. So there are several columns. The first one is where the enrichment was done. So we know this is the ANOVA significant column, where everything was significant. And in this output table, these are only the hits that are statistically significant according to our threshold, right? So everything in here is theoretically interesting. So you see the type, which could be biological processes or cellular compartments and everything else that we had in there. Here you see the exact term that is enriched. Here you see the numbers that were used to calculate this contingency table in order to perform the Fisher's exact test. And the really relevant numbers are the FDR. So again, this gives you an idea of how confident you are in finding this hit. And the enrichment factor is the other important measure, because you would see values that are larger than 1. In this case, this means that this category is enriched among the significant hits. And if the value is smaller than 1, this is something that is depleted among your significant hits. Now, I said that this is not the best way to do the enrichment analysis. Any ideas why? Or what kind of information are we missing in this case?
I think we have filtered before, so not all of the proteins which we actually have measured are in this.
I didn't filter, I kept the entire matrix.
No, for valid values.
But even that is okay, it doesn't change anything. Other guesses? That is also not, that is okay. I mean, you can't do statistical analysis unless you have enough data to do it.
And maybe significantly up or down regulated?
Exactly. So in this case, we're just looking at everything that is significantly different, but we have no idea if it's up-regulated, so if the expression is going up or down. And then, of course, we're maybe looking at different processes, some of which are going up, others of which are going down. So we lose all of this information. There's another thing that we lose. So in this case, we're comparing 3 groups. And our output with this significance, it tells us only if a protein is different between one cell line compared to any of the others. But we still have no idea where this difference comes from. So what do we do in this case? We are going to go to the previous data matrix. So this is the one immediately after the statistical test here. And we're going to take only our significant hits for a second. So to do this, we'll use the filtering options that Tami demonstrated in filter rows. We'll filter based on categorical column. And in this case, we want to filter by significance. So we'll take everything that is marked with plus, but in this case, we want to keep these matches. So we'll change the modes. We don't want to remove this. We want to keep the matching rows. So keep everything that is marked as significant. OK. So what is really visually very informative is to just cluster all of these significant hits. But before clustering, it's always a good idea to normalize the protein expression. So a good way to do this is just z-scoring the data. That is, I was a bit fast maybe, that is in normalization here, and then z-scoring. We keep rows because we want to normalize across the different cell lines, just to make sure that there aren't proteins that have really larger differences between the cell lines, but they're kind of on comparable scale. We don't want to change anything, so we just click OK. And now we can try to cluster our significant hits and then see what are the patterns in the data that we see. So here there's the clustering and PCA menu. We can choose hierarchical clustering. There are, again, a lot of parameters here that allow you a lot of control over the way the clustering is done. I think I'm not going to go into detail in all of these, but as Jürgen mentioned, it's possible to cluster both by column and by row. These are all the usually specific parameters for hierarchical clustering, so the distance, the linkage, and the constraint parameters. But the one that has the biggest effect is probably the distance measure, but in this case, you can just keep Euclidean and you don't need to change anything. But in case you have really very different data or it doesn't work well for you, you can just try and play with other options, such as Pearson or Spearman correlation, and then this would be more relevant, for example, if you would have like oscillating data or data with some specific temporal pattern. But these are really very special cases. The same is true for the column clustering, and then you see there's one more option, which is pre-processing with k-mean. So this is an option to help you to cluster the data when it's really enormous. So this was the problem also in the past when we didn't have such fast computers. You can use it or not. It will not change much in this case because our data is not very large, but it's really a lifesaver when you have huge data. So again, we don't really need to change anything here. These are all the columns that will be used for clustering, so automatically selected just all of our main columns, and we can just click OK. You see that it's really super fast. It's already there. OK. So the first thing that you see, and this, of course, you would expect, so it's not a big surprise, all of our biological groups are clustered together. All of the replicates are clustered perfectly. And you also see here that these are all the proteins that, that lead to this clustering. There are already patterns in the data. So you can see that there is a cluster that is high, that is highly regulated, or there's higher expression of this group of proteins in this cell line. Then there is another cluster that is highly expressed in this cell line, and so forth. But there are many more things that we can do here. For example, what I like to do is to change the colors. of the heat map, especially for those people who can't really distinguish between green and red. So you would click on this icon here, and then to change the color, you just click on the particular, what is this, an arrow, and you can select any color. So blue is usually nice. You can add additional colors just by clicking anywhere you want. You just click, and then you can just add another color. And you can also remove it by right-click. If you want to change the min and the max value, you can do it again by right-click in this area here. And you can modify this if for some reason you want to change this. I'm not going to change anything here. Right. So we can also change a little bit how this is plotted here. So the length, for example, of the dendrograms and the distance by clicking the communist button, as it's known internally. So you can modify the row clustering distances again. So for example, we can decrease a little bit the width here to, I don't know, 200 and 100. Maybe remove this draw to width, but you can really play with this depending on how you wanted the plot to look like. So you see that now this just got a little bit shorter. And you can do the same for the column height. So maybe make it 170. Okay. So another thing that is helpful is that you can define the names of columns and of rows. Let's start with the columns maybe. So configure column name. You can either use the names of your samples, or you can use the grouping. This can be useful, again, if you have many, many samples, and then it will be difficult just to see what the names are, but you can instead use the grouping. Or what you can also do is you can set a column color bar. And again, here you can choose the grouping. So let's do this. The column color bar specify that this is our experimental design still. And what happens is then you see that this color is added. And again, in this case, doesn't help much, but if you would have 500 samples and you have no idea if your clustering worked well or not, this is really easy to visually inspect how well the clustering worked. So with respect to the rows, it's very similar. So you can configure the row names. And you can use pretty much anything that is already in your data. You can use gene names. You can have additional identifiers, like protein names. Or actually, in this case, remove it to shorten it a bit. And we could also add row color bar if we would want to specify the clusters, but we don't have to do it. So we just remove the additional row names here. Click OK. Now, if we want to see what are the proteins that actually form these clusters, we can do this on this plot by zooming in. So to do this, you just click anywhere randomly, and you zoom in. So with the mouse, it's just dragging it, and you have to keep zooming and zooming and zooming. Okay, so you see that something starts appearing. We have quite a few, but you get the idea. So they're all in there. You can then, once you have zoomed in, you can actually scroll up and down to navigate. Okay. And to zoom out, you can double click again. Okay. So the next thing is that maybe you're interested in actually how many different clusters you have with respect to the protein. So what are the clusters that really distinguish between your 3 different cell lines? And you can do this in 2 ways. One would be define row clusters here. And you can specify how many clusters you want to get highlighted. So you can try to visually kind of get a feel for it. Let's say you see 1 here, 2, 3, let's say 5 or 6 clusters. You can just increase this, say 5. And then if you click on apply, all of these clusters will be automatically highlighted on the dendrogram. So yeah, you can increase this, decrease this, anything that makes sense to you visually. And then you will see in the field next to it, here in the upper panel, row clusters, that these different clusters are also kind of predefined here as well. And what I mean by this is that if you click on this one, so this corresponds to this cluster here. And you can see that there are 748 proteins in this cluster. This is some random number that is associated to identify this cluster. Yeah, really can just be used as an identifier. And another thing that is very helpful, and maybe you have seen it in a lot of publications, is this profile plot here, which is a heat map. So you really nicely can see in a 2D format what is the pattern of this cluster. So you also can see it here because we know that red corresponds to high expression, but then you can, it's kind of more natural to see it in this pattern. So yeah, everything is high here. You can also define the colors here. So this heat map, at the moment, everything that is close to the center of this cluster has a hotter color, so to speak, and everything that is further away, cooler color. You can then look at the different clusters. Sorry, I clicked 2 now, so there's no clear pattern. Of course, you can export all of these as images. You can export this cluster. You can export the heat map, again, in any of the formats. So what is interesting is that you could also do the enrichment analysis on this level. See a question? Many questions?
When exporting, especially the heat maps, is there a way, if I export it as a PDF, to not lose the whole annotation? Because it only exports what I actually see.
So actually, I tried it earlier, and if you zoom in, for example, on a particular region...
Only if I zoom in, yes.
Yes.
If I can actually see it here, I also have it in a PDF.
Yes.
But if I want to have the whole heat map with the whole annotation...
You mean with the protein names?
Yes, yes.
Yeah, I think if there are too many, then it's difficult. So it's in there, but it's, yeah, it works if you zoom in. If you have fewer and you have the names, you can then export it with the names. I think if you just want to have the whole thing, it's also on what kind of, if you just imagine, right, if you have 4,000 of this, what kind of font would you need to have to place it on an A4 paper?
Sure, but I think the cutoff is already at the 100 or so.
I think with 100 it should work. I think for at least for 30 or 40, I tried it earlier and it worked. So yes. Okay, so the limit is between 40 and 100. Okay, but I think that is maybe something that the developers can look into, because yeah, that would be the way to really zoom in and then get out that figure.
If you go, it's just
I will come to you just.
Hi. Is there a way to define the optimal number of clusters?
So in this case, it's difficult. Like, what is the optimal number? It's more up to you between how you want to distinguish and between which groups you want to distinguish. So in this case, it's entirely up to you. and right, because maybe you say in this case it's cell lines, so, but if it was more really looking at different subtypes of a disease or disease versus health, you would really know that you want to contrast these groups. So then you would search for kind of clear clusters that are very distinct between these two groups.
No, I meant in the protein, the same.
Yeah, in this case, yeah, there's no optimal number, yeah. Oh, I see one more question at the very back.
Oh, just wondering how NAN values are handled in terms of the t-test, like when it's calculating mean, variance, that type of thing.
So in this case, in Perseus, you need to have at least 3 valid values for the test to be performed. So any group that doesn't have this will just be ignored in this case. It will not be used for the test calculation. So they're just not used, yes.
Okay, is there possibly another reason why if you use the same data set but you can conduct the ANOVA in log scale versus like raw LFQ values, you can get different number of significant different
Actually, you shouldn't use it on the non-log data because in our case, what 0 is, it means it's not detected. So it's not a real value. It doesn't mean 0 expression, but rather not detected. So you don't want to do this, yes. So you will get definitely different results. Now, I should say if you're interested in pull downs or experimental setups that are conditions that are very different, that's a separate story. So in this case, we would recommend, again, to log transform the data, but you would have to use imputation in this case. And I'll talk about it tomorrow again, because exactly you do not want to lose these cases, but you want to log in order to have the right distributions, which is why you need to impute then this missing data in a way that is going to allow you to perform the test, but then not introduce false hits.
Okay, thank you.
Okay, so I mentioned that there is one more way in which you can do the enrichment here. So for example, if we select one of the clusters, as it is here, so you can click on one here. What I didn't mention also is that you see when you mouse over certain parts of the dendrogram, there's a little square that appears. So if you would click on this, then you can define new clusters. Or you can click on the one that is already predefined, but then that's a new cluster, and this would appear then here if it wasn't there. And you can really choose anywhere else, so you can kind of manually define clusters. But we'll just select one from the table, any, and we'll check. So we also know that this is a cluster that has high expression in the first cell line compared to the other two. And we'll check if this is characterized by particular goal terms or KEK terms. To do this, there is this little button here called, or it's not called, but it just says, the description says show enrichment, so we can click it. And it will again calculate what terms are enriched in this, compared, however, only to what we have in this filter data set. So this is why this is again a suboptimal way of doing the enrichment, because you should always remember against what background you're enriching. And in this case, we have already filtered only for significant values. So we're really enriching only against these proteins that we have in here. But it can still give you some idea of what kind of processes might be really different among the significant hits.
We saw a lot of people have issues with the clustering, that they have only one huge cluster and they cannot have more than one. So don't worry. The issue here is that the computer is confused about their language settings, so that the number has a comma and not a dot. So once you change your computer to English settings, you will be able to do this, but hopefully we will fix this for the next version.
Sorry for this.
Okay, so I said that is a suboptimal way of doing the enrichment, so what is the best way to do the enrichment? We still want to keep this information about what is going up and down. We still want to know what are the clusters that are different between which groups. So now we know that these are kind of these clusters that we have defined here. They're sort of cell line specific. So for example, we know this one is high in this cell line, this one is high in this cell line, and this one is high in this cell line. So we want to keep this structure in the data. So to do this, we can just export row clusters. Click here. What this does is then just keeps the old table, but there is an additional column here. This additional column is called cluster, and you see these random numbers, which were the identifiers of each of the respective clusters. So now we know that this is the first cluster here. Scroll down, and then somewhere down should be the other clusters. Yes, you see this is another cluster. Okay, so kind of have this information. So now we can, again, go and do our Fisher's exact test. So in notation rows, Fisher exact test. And in this case, we're not going to look at the only at the column that says, okay, this is a significant hit without caring about the direction of the expression, but we're going to take our cluster column. Because in this case, it will be very similar to the enrichment that we just did in each of the clusters, but in this case, we'll take the correct background. So we'll take the entire matrix that we have used in the data analysis as a background. And then we don't need to change anything else. Just click OK. Oh, sorry. Actually, that is wrong because I just exported the matrix. I realized, so here, where we did the test, we still have only our significant hits. So I apologize for this mistake. So we'll remove this. We can just select this and just remove them with the X here. Okay, so in this case, we still only have our significant hits. And we want to make sure that we have our complete table with all the proteins that we used for our enrichment. So to do this, we need to go to the table before filtering for significant hits. So remember here we filtered. You can also check if you're not sure on each process, you can click. And then you can, if you open this last hidden window, you can see, first you can see what process you applied. So filter rows on a categorical column. You see the time that it took to apply this process. And you can see exactly what you did. So it's a bit difficult with this resolution, but normally it's easier. So you see that we actually filtered for the Tyanova hits. So we want to go to the table before this. In my case, this is matrix 17. And what we'll do is actually we'll match two tables. And this is something that is very useful in many other cases where you really want to bring together two tables. So this could be two proteomes, proteome, transcriptome, or anything else where you would want to kind of either get both or just transfer certain columns from one table to another. So to do this, we can select this matrix, matrix 17, so the one before filtering for significant hits, go to multiprocessing here, and then select matching rows by name. So what you see here is that we have to define two tables that we want to match. The first one is base matrix. As the name implies, this will be the matrix on which data will be transferred. So we already pre-selected, this is matrix 17. We need to define the other matrix. So to do this, we have to select it in this table. And then from the workflow, we will click on the table that we want to match. So this is the one in which we exported the clusters, or the last one in your workflow if you deleted all the other nodes. So just click on it and you'll see it's updated here in the match by name. You click OK. There's a bigger menu then that starts asking you how exactly to do the matching. So in order to be able to do this, you need to have the same identifiers in both matrices. In this case, it's easy. You can just use the uniprot because we know that they're kind of, one of the tables is just a subset of the other. So we can just keep this. And the only thing we want to do is to transfer this cluster column that we exported from the second matrix to the base matrix. So we just select the cluster because everything else we already have in the base matrix. We select the cluster and we just click OK. Okay. So now you see we have the larger table. So this is now the full set of proteins that we had. And we have transferred our cluster column as well. So this is the table in which we want to do the enrichment finally. So now we can go to annotation columns again, the Fisher exact test, then select the cluster column. We don't need to change anything and just click OK. Sorry. Right. Was this clear about these 3 different ways of doing the enrichment, and then what are really the differences and the benefits and the outcome? Okay. So you see it's the same table that we got before, but then you realize that now you can actually check in which cluster significant term was enriched. So you would have to kind of keep in mind the naming. So for example, if you're looking at this particular cluster, it has this ID 2633. So you know this is high in this cell line here. Then you can go to the output, sort again, find the cluster that you're interested in. I think it was the 3.3, and then now you see that this will be characteristic for, like the terms would be characteristic for this cluster, and you know exactly what is the pattern of expression of proteins in this cluster. But the rest is really the same, except for now you would have the enrichment for all the different clusters here. Okay, one more thing that is also very helpful in terms of analysis, although you maybe usually want to do it at an earlier stage, is the principal component analysis. So that's a completely different topic. And this we could do, for example, at this stage. So we should not normalize our rows before principal component analysis. So we're going to go back to the table before the z-scoring here. In this case, matrix 19. And downside of principal component analysis is that actually we cannot do it when we have missing values. And you remember that actually, or you don't have to remember, you can still see that we have some NANs here and there. So the first thing we have to do is in this case to get rid of the missing values. Of course, we could in other circumstances impute those and still go ahead. In this case, we're just going to filter them. So it's again the filtering menu. Filter rows based on valid values. And to remember that we have 3 by 3, we have 9 samples. So we're just going to require that in all samples there is a value. Click OK. And now on this table we can apply the PCA. This is together with the clustering. So in analysis, clustering PCA. Can select principal component analysis. And there's just one additional property we could tick in here, and this is category enrichment in components. So this is yet another place where you could do enrichment. But in this case, the enrichment will be applied to the components of the PCA. So let's just do it and then see what happens. There are more parameters. You do not have to change those, but here you can define in which components of the PCA the enrichment can be done. It's usually the first most significant, or rather the first more important ones. And you can increase or decrease. So if you increase it, there will be more computational time that will be taken because more components will be checked. Again, the FDR is applied here, and we're not interested in the relative enrichment at the moment. So we just don't change anything, click OK. So usually principal component analysis is something that you may want to apply at the very beginning of your analysis before you start any statistical analysis. And why this is a good idea is because there you can very easily see if there is a sample that is different from the rest of your sample. So usually you have your different groups and you expect them to be combined together on the plot, but you can easily spot that one of your samples is really off in there, if this is the case. So it's a little difficult to see here, but this is the output of the principal component analysis here. So these are the 9 samples that we have from the 3 cell lines. And what you see is that they already are nicely grouped into 3 groups. And you can just start guessing if these are our, these correspond to the 3 cell lines, and the answer is yes. So if you would increase here the size, you can just click on each of the samples one by one and then see where on the plot this sample is. Then you can select the first 3, let's say, and then you see this is one of the groups. You can, similar to the scatter plot that Tommy was showing, it's really the same functionality, so you can change the color of each of the dots. You can change the symbol type. You can change the size, and you can also add a label to it, just for clarity. So you see that they're all here. You could also, it would be faster if, since our groups are already preset, the grouping is then available under categories, so you could select all of them. And then click on these multiple colors, which automatically then gives different colors to the 3 groups. And then you see that they're exactly perfectly grouped in the PCA. Yes, you could, in theory, you could zoom in and zoom out, but with 9 points, we really don't need to do this. You can export this. And what is really interesting in this case is to look at what is called the loadings in this case, which is in the lower panel here. So these are actually all the proteins that directly correspond to the upper panel, that correspond to driving this difference among the different groups. And they're actually like a mirror image to the sample separation that you see above, which means that we know that currently component 1 is the component that really very well describes the difference between the 3 cell lines. So it accounts for more than 60% of the variability, which means that then the proteins that are kind of spread horizontally are the ones that are driving this difference. And here, very naively, we can just look at what those proteins are. Again, similar by using this rectangular selection. You can just select several proteins. Okay, here you see some strange identifiers, but you can change what you see in the label type. You can select gene names, for example. And then you see, you know that these proteins are really very differentially expressed between your groups. So it's a very easy way to kind of already spot interesting proteins. Now, because you can't read nicely the labels here, there is a nice trick that you could use. So all the proteins are still listed in a table here. Which is the lower table here. And the nice trick is that if you right-click, so you have selected some proteins here on the plot, you can right-click and then select this option, bring selection to top, which is just going to bring all of your proteins on top of this. And then you can actually see what's behind it and what the names are. Okay. Then you remember we did this enrichment step, this additional step to this analysis. And the result of this is in this category tab. So what this does is that for each of the first 5 components, because this is what we selected, again, different categories are checked for enrichment. And we also know from the PCA that component 1 is the one that has the largest amount of variance, so this is the really interesting one. And we want to know what are the categories that are in there. So here we can just sort, look at component 1, and then check, very similar to what we saw before, what are the terms that are enriched. Here. With the FDR and with the difference again. And you can, again, if you select a certain category, you can see where it lies on this loadings plot. So I don't know what is a bigger one, but what you see as a field is that usually this is a group of proteins that is kind of located towards one of the ends of the plot on the respective component. So you see that this is more going towards smaller values of the component. Okay, so I would like to allow a couple of minutes for questions. It can be really with respect to anything that was shown now or anything that was not shown.
So, hello. I always do label-free quantification, and I always apply normalization in MaxQuant. But as I said yesterday, I can still see some batch effects despite the normalization. I wonder, is it okay, really, to perform a second step of normalization in Perseus?
So, can you describe a bit what you mean by batch effect?
For instance, I mean, I have 2 conditions, but they were cultured at different times. So batch 1 and batch 2. And what I can see in I make this multidimensional scaling plot, similar to the PCA plot. And I can still see that there are batch effects. I mean,
Okay.
sample from sim.
So why I asked is because in many cases, you may expect that you would have batch effects, and then it's really important that you do your experimental design in order to account for this already. So, okay, in this case, maybe you can't if you're constrained by, I don't know, your collaborators. That's why you do it in different time points. But normally, the best way to account for the batch effect is already at the experimental design level. Now, if this is not possible, you can do it at different levels of the analysis, and actually you have to do it before you start any statistical analysis. So standard ways are what Tommy showed, like just subtracting median value from all of the samples. That is already going to help. What you can also do is, this is actually a very nice trick. You could do the principal component analysis, and if, let's say, you see that, so in this case, we see that everything is nicely grouped already in our first component, which means that we see biology at the very top level. But in your case, if you have a strong batch effect, your samples may be nicely grouped according to these time points. That is not what you're interested in. So what you can do in this case is you can actually subtract the component that accounts for your batch effect. So this is another way in which you can normalize your data. If, of course, this is very important that it is decoupled from your biological variability. So that would be another way to normalize it. But in any case, yes, you have to, if the batch effect is your driving factor. Okay.
So in the enrichment analysis that you're doing based on the clustering and the previous clustering, how would the fact of that you are choosing how many clusters you want to do, how would this affect in the enrichment analysis you are like doing afterwards? So for example, instead of 5, if you put like 10, and then you do the enrichment analysis, how would these two enrichment compare between each other?
They will be different, right? Because you're looking at a different set of proteins. That's why it's in the first step, when you define the clusters, you need to have some rationale as to why this is the cluster that you're interested in. So of course they'll be different. It's completely different proteins you're looking at.
Hello, could you explain a bit about volcano plots that we
Okay.
just some few?
Anything in particular?
I don't know how to
Just how to do it. Okay.
how to do it, what to extract. I mean, how
Okay,
to-
so for the volcano plot, it actually works when you're comparing two conditions. So let's go ahead and do this. Okay, here we did the multiple test. So if we just use any matrix, actually it should work with any matrix where we have the groups defined. We could go to this miscellaneous. This is where the volcano plot lies. And again, we have to define which groups we want to compare. So in this case, it comes from the grouping, and then let's say it's these two cell lines. So these are the two groups. Then we have the type of test. It's t-test, sidedness, number of randomization. So it's very similar to the Tyanova that we spoke about, preserving groupings only if you have technical replicates, FDR, and the S0 parameter. So don't need to change anything. And it's already there. So yeah, you see that right now, because this S0 parameter is really very small in this case, so almost everything appears to be significant. So here you can actually change this value, I don't know, to 2, let's say. And there is a refresh button here, these two green arrows. So once you change one of these two parameters, either the FDR or the S0, you have to refresh the plot. And then you see immediately the effect of changing the S0 parameter to the hits that are significant. So what you just saw is that when we increase the S0 parameter, essentially we're asking for larger fold change difference, right? Now you see that the hits that were significant before here, with really small difference, because on the x-axis we have the fold change, they're not significant anymore, but in this case we include more points here that have worse p-value, but larger fold change.
Yeah. I have also a question. How can I show the data in form of a volcano plot after the multiple sample test?
After?
After I have done the multiple sample test, meaning the Tyanova.
Yeah, then you would still have to go for each by each for pairwise comparison. So
Yeah. You would normally do the post hoc test? Or?
yeah, okay, that would be another option, yes, of course. So one option was the clustering, where you could define the difference between each of the pairs or between the different groups. And if you really want to know between which two groups the differences are, there's also the post hoc test that is implemented in Perseus.
Yeah, and the problem is with the previous version I have or I use, that then it's not possible to make it visible in a volcano plot because there's missing any p-value or so for the y-axis.
Okay, I would then again advise to check the latest version if there's still a problem, and if the problem still persists, to contact the, use the complaint form or the bug form.
Okay, good, thanks.
Hi, so I don't know, maybe this is a question for the developers, but how does Perseus calculate the difference, the log of the fold change? Is it the subtraction of the mean of the logged values, or is it the log of the ratio between the mean of the intensities or the counts?
So it's the log of the difference between the means.
But if you are doing the tests, you already have the log transform data,
if it's
right?
the log transform, then you will just take the difference between the means because they're already logged.
Okay, and does it?
You mean about the fold change here, right?
Yeah.
Yes.
Will it make a difference then in your volcano plot, like the proteins that show as differentially expressed, if you use one or the other?
if you do it in the right way, no, right? The important thing is that you need to know that if your data is locked, to take the fold change, you don't divide them, but you subtract the two values, right? And if it's not locked, of course, you would divide them. So,
okay, so I can apply different formulas in this case to different kinds of data, and they will be comparable.
yes, as long as you follow the rules of mathematics, yes.
Okay, thank you.
Okay, so I don't see more urgent questions, but I would want to say that also in there is the exercise sheet of today. You would find it, so this is in the USB stick assignments. So actually today we did this P2 Perseus.
Stefka, sorry, one more question.
Simple. So, what is the rule of thumb for S0? How big should I choose?
So we actually advise no rule of thumb, but rather look at the data and then decide what makes sense for you. There is a way to actually calculate a fixed S0, but then in the end of the day, it's kind of up to the users to decide what is really the best one visually by just playing and then checking what is the final outcome of the test. And you should always remember that it's kind of a trade-off between the fold change and the significance value, and then you need to know what you're comfortable with. Okay, so back to the previous part. So there are actually 4 tiny exercises in the Perseus sheet. We covered the first one today, which showed all the main functionality in Perseus. There is then there is a second data set, which is label-free data. And you can follow again the steps that are described here, but it essentially asks you to repeat the same steps that we did now, but with label-free data, and to answer a small question, but it should be very easy for you after this. There is another exercise on supervised learning or machine learning with Perseus. Again, you can follow the data. So here there is a breast cancer data set with different subtypes, and the goal is to use the machine learning module in Perseus to find proteins that are really, again, different between the subtypes, but that can be used for something like biomarkers, though that's really a big word. This is something that probably a lot of you saw yesterday that was covered in the bootcamp, but the data is in here, the exercise is described, so you could just repeat it at home. And there is another exercise that is actually analyzing NGS data. And I also urge you to try this later at home. And the really interesting thing is that here you could use transcriptomic and translatomic data and actually load the data, calculate RPKM values, following the steps here, and you can actually check what is the relation, or rather the correlations between these 3 levels of transcriptomic, translatomic, and proteomic data, and to see if this is what you would actually expect. But all the data and the descriptions are in here, and if you have questions, you can always ask any of us later. Okay, so if there are no more questions,